Hello, biology students. Welcome one more time for a virtual lesson on DNA structure and function. Today's lesson, the final installment in the series, is on transcription and translation from both sections 8.4 and 8.5. And this deals mostly with how does DNA actually work. We know the structure of DNA, we know how the pieces fit together, we know how it copies itself, but how does it work? How does it actually do its job to control traits? And that's where these two processes came in. One of the people who was really central in figuring that out was Francis Crick, the guy who first figured out the structure of DNA. And he had identified something called his central dogma, in which he figured out how DNA works. What Crick said was that it goes from DNA, and that translates into RNA, and then that in turn makes proteins. So the information in DNA goes from the DNA to the RNA and then finally into the proteins. It's the proteins that give you your traits. Whether you have brown eyes or blue eyes or green eyes depends on your protein that's found in the eyes. He called this process of going from DNA to RNA transcription, and the process of going from RNA to proteins translation. Um, One thing he found is that RNA is the messenger. It's the go-between between the DNA and the proteins. And the proteins, of course, are made in the rough ER out in the cytoplasm, and the RNA is the messenger that carries the message to that place. So let's talk about the process of transcription. It's actually quite similar to replication, where the DNA copies itself. First thing that has to happen is the DNA has to unzip and expose the bases at the site where the gene that needs to be read is found. And so it unzips itself at that location. Free-floating nucleotides are going to attach themselves to the exposed DNA. There's a different enzyme this time though, so then rather than making new DNA, it's actually going to be making RNA. The RNA strand will be completed. When it does, it will detach itself. The enzyme will actually detach it from the DNA molecule, and then the DNA is free to zip itself back up. And this process can be repeated whenever more RNA is needed, whenever more proteins need to be made out in the cytoplasm. It'll simply unzip that gene again, make another copy of RNA, and send it back out to the cytoplasm. And so the DNA is kind of like a book that can be read over and over again. The RNA is like a working copy that when it's done, it's actually destroyed and thrown away. So let's talk about this molecule RNA. It's like DNA, but it has a few differences from it. So unlike DNA that has a double-stranded structure, RNA is a single strand of nucleic acid. RNA doesn't really have a true helical shape, although it is kind of a spiral. It tends to kink up and form other structures, and it's not a true helical symmetrical structure. It uses a different base. Rather than thymine, uracil is going to, or RNA is going to use the base called uracil, and it's structurally very much like thymine, but it is different. And so in RNA, the U is going to pair up with the A instead of the T. And the last thing that's different between RNA and DNA is the sugar in the RNA is different. It has an additional oxygen, so it's not deoxyribo nucleic acid. It is simply ribonucleic acid because it's got an extra oxygen. There's two different types of RNA that are made. One is called mRNA, which stands for messenger RNA, and one is called tRNA, which is transfer RNA. mRNA carries the codes for making a protein out to the rough ER, and the tRNA goes out into the cytoplasm, finds the amino acid that's needed to make a protein, and brings it to the mRNA, the site where proteins are actually going to be made. And that process happens in what's called translation. Translation is reading the codes that are encrypted on the RNA, figuring out the amino acids that go into making the protein, and assembling them in the correct sequence along this mRNA strand. So how does this process work? So first of all, the exposed bases of mRNA um, are on the rough ER. The tRNA brings the amino acids and it attaches to the appropriate site on the mRNA. It's kind of like finding the place on a puzzle where a piece goes. So they find the proper place, they attach themselves there, and they all bring one amino acid to this growing chain, which will eventually be a protein. When all of the tRNA has brought all of its amino acids there, in the end we will have a protein. A ribosome will come along, it will detach that amino acid chain, and in the end the protein is complete. It is this protein which will control the traits, and that is how really that your individual traits are different from other people's. Your DNA had codes. Those codes were translated into RNA, and then those RNAs were read in the process of translation by the transfer RNA and the ribosome. In the end, you'll have your proteins that make you you. And in the end, that is the whole process of reading the codes in DNA, and that was discovered a lot in great part by Francis Crick. One of the things that we have done since Crick's discovery of that was we figured out what all the different sequences of bases mean. We found that DNA and RNA are read in 3 base sequences called codons. And the different letters in different order, like CCG or something, has a specific translation. What it means, CCG means that you're going to put an amino acid called proline into the protein at that point. There are also other codons called start and stop codons, which basically tell you start the gene here and stop the gene here. And so that tells you exactly how long the protein is going to be, how many amino acids to put on, so it doesn't just keep building and building into the future. And once you know these codons and what they mean, we can actually start to translate pieces of DNA and really learn the secrets of what they're hiding inside. So there's still some exciting research to be done, figuring out, knowing codons, figuring out what that protein will actually do when it's made. And we're not there yet, but there are definitely some progress being made that way. And all that's due to understanding the structure and function of the DNA molecule. This is going to conclude our virtual lessons for Chapter 8. I hope you enjoyed the lessons and hopefully they're helpful. Good luck on the test when we get to that point.